Well, hello. Thank you for joining us today. We'll get started. I'm Tracy Smith, the Collaboration, Education, and Outreach Coordinator for the Center for Integrated Cellular Analysis based here in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we will discuss PerturbSci-Kinetics, a novel combinatorial indexing method that captures single-cell transcriptome, nascent RNA, and sgRNA identity in pooled CRISPR screens. As always, we'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. And our guest today is Zihan Xu. Zihan is a PhD student in the Cow Lab at Rockefeller University and received his Bachelor of Medicine degree from Shanghai Jiaotong University School of Medicine. His research focuses on developing new multimodal single-cell technologies for studying RNA temporal dynamics and gene regulation in various biological contexts. With that, welcome. I'll hand it over to you.
Thank you so much, Tracy, for your kind introduction. Can everyone see my screen now?
Looks great.
Okay, then I will start. Hi, everyone. I'm Zihan, the second-year graduate student in the Cow Lab of the Rockefeller University. It's my great pleasure to be here to share our research. Today, I would like to talk about PerturbSci-Kinetics, our newly developed method combining the metabolic labeling and combinatorial indexing single-cell CRISPR screen to study regulations on RNA dynamics. CRISPR screen has given people the unique opportunity to study the functions of genes. For bulk screen, individual guide RNAs targeting different genes were transduced to different cells expressing Cas9, resulting in the loss of function of different genes in different cells in a pooled manner. After the screen, we could extract the guide RNA sequences from the bulk pool, and by comparing the guide RNA abundance before and after the screen, we could infer the essentiality of genes in different biological processes. However, to study the functions of gene regulation, we need to establish the linkages between the genetic regulators and downstream transcriptomic responses. So transcriptomes are needed in addition to the growth phenotypes offered by the bulk screen. Recently, people have successfully combined CRISPR screen with single-cell RNA sequencing. And by using these excellent techniques, we have been able to obtain the single-cell whole transcriptome phenotypes after the perturbation and use this result to establish the gene co-regulation modules. However, currently there are two main limitations. First, all these techniques are built on the microfluidic system or even sorting-based system, which largely limits the number of cells that we could process. Besides, gene regulation is more complex than just a transcriptomic snapshot. It has been a common sense that the mRNA levels of the genes are tightly regulated by the transcription. However, gene expression is a dynamic multi-step process. mRNAs are transcribed from genes encoded by DNA. And after their missions are accomplished, these molecules are degraded. Assuming we detected a gene with upregulated mRNA level, which means the steady state upregulation. So in fact, there are two possibilities. It could be due to the enhanced transcription mediated by the binding of transcription factor to the distal or proximal sites, or it could also be due to the reduced degradation, which could result in the accumulation of mRNA molecules in the cells. But conventional gene expression assay cannot reflect this dynamic process. To address this issue, a smart strategy called metabolic labeling has been invented to track the nascent RNA. For example, by incubating cells with 4SU, a uridine analog, the cells will efficiently uptake this analog and incorporate them into the nascent RNA. And after the RNA extraction, we could perform the chemical conversion to convert 4SU into another form, which would be recognized as guanine during the reverse transcription. After library preparation and sequencing, we could computationally retrieve the origin of each read by using these artificial T2C mutations. By combining the metabolic labeling and computational modeling, the RNA synthesis and degradation could be inferred. Besides, metabolic labeling has also been compatible with single-cell RNA-seq and has been used to study the cell state transition. So, to enhance the scalability of single-cell CRISPR perturbation and to study the gene regulations at the RNA dynamics level, we developed PerturbSci-Kinetics. This is the overview of our technique. We transduce a guide RNA library to the culture cells and then induce the CRISPR interference to knock down different genes in different cells. At the end of the screen experiment, we added 4SU to the culture media to label the nascent transcriptome of single cells. About the CRISPR screen, we use a strong Cas9 enzyme called Cas9 Crab MECP2 to maximize the knockdown efficiency. And for guide RNA expression, we adopted a modified version of crop-seq vector, which could generate both short functional guide RNA and a long transcript containing the guide RNA sequence for targeted capture during the library preparation. After cell fixation, we could perform the chemical conversion to convert 4SU. Then index reverse transcription was performed using the oligo-DT primers, targeting the poly-A mRNAs in the cells, and another targeted primer capturing the guide RNA sequences. Index ligation and indexed multiplex PCR were performed to add the second and the third indices onto cDNA molecules of single cells. By using another round of guide RNA enrichment PCR, we could further separate the guide RNA library from the whole transcriptome library for sequencing. Based on EasySky, our platform is fully compatible with million-scale single-cell CRISPR perturbation profiling in one library preparation. And even in the current small-scale format, it has been more than 60 times cheaper than 10x PerturbSci. So first, I would like to show some quality control data. We benchmarked the whole transcriptome capture of our PerturbSci with EasySky and saw almost perfect correlation. And we validated our DOX-inducible system at the single-cell level. DOX treatment could activate the expression of Cas9, and it successfully knocked down the target gene. And by mixing human HEK cells expressing a human guide RNA and mouse 333 cells expressing a mouse guide RNA, we performed a two-dimensional species contamination assay. And we plotted the percentage of human whole transcriptome UMI and mouse and human guide RNA UMI detected from each single cell. You can see for cells with mostly human transcriptome UMI, they are mostly with the human guide RNA. And for mouse cells with almost no human reads, they are mostly with mouse guide RNA and at the lower left corner of the plot. Both populations showed correct and very strong knockdown effects. We further tested the compatibility of 4SU labeling with PerturbSci. By mixing control cells, 4SU labeled cells, and 4SU labeled chemically converted cells into one library preparation, we could only retrieve the nascent transcriptome from labeled converted cells, indicating the specificity of 4SU labeling and chemical conversion. And by checking the exonic reads ratio of reads of different origins, we could see the clear difference. For nascent reads, they are largely mapped to the intronic regions, indicating the labeled RNA are largely unspliced and newly synthesized. And for pre-existing reads, they are mostly mapped to the exonic regions, indicating they are mature mRNA. Chemical conversion requires additional chemical and relatively high temperature, but still, after this step, we could detect decent guide RNA UMI counts from single cells. And the chemical conversion step did not cause doublet formation nor severe cell clumping during the library preparation. So these results indicate the success of PerturbSci-Kinetics to capture the whole transcriptome, nascent transcriptome, and single cell, single guide RNA identities in single cells. So we selected 228 genes, including known transcriptional and post-transcriptional regulators, essential and relatively non-essential genes, and performed our proof of concept pool screen experiment. It's clear that this perturbed pseudobulk transcriptomes are clustered by their function in the UMAP space. For example, For this cluster, these genes are members of nonsense-mediated mRNA decay pathway. And these genes encode RNA exosomes. Transcription-related genes are here. These are RNA splicing genes, and these are mitochondrial genes. We also saw significant reduction of the expression level of the targeted genes. Beyond that, by using the nascent transcriptome data, we could further infer the synthesis and degradation rates of these targeted genes. So you can see only the synthesis rates of these genes are significantly suppressed, while their degradation rates did not, compared to non-target cells. This result, which matches the functional mechanism of CRISPR-I, and it also supported the feasibility of our computational modeling. After the development of this technique, we seek to address the following biological questions. First, by using this technique, can we identify master regulators of global synthesis and mRNA degradation? So we calculated and compared the distribution of global synthesis rates and degradation rates between perturbations and non-target cells. The volcano plot showed the relationship between the significance of the changes and the extent of the changes. And the bar plot showed the fraction of significantly perturbed genes in each functional category. Overall, we saw more transcriptional regulators than post-transcriptional regulators, and we did not observe any post-transcriptional repressor in our screen experiment. More specifically, knocking down chromatin remodelers only resulted in the changed global synthesis rate, but has no effect on global degradation rates. And mRNA decay-related genes are relatively more overrepresented in the list of genes that cause global degradation rate changes. Very interestingly, we found knocking down AGO2, a core component of microRNA-mediated silencing complex, could result in the increased global synthesis rate. The start of transcription consists of three phases: transcriptional initiation, pausing, and elongation. Transcriptional upregulation could be driven by either more active recruitment of RNA polymerase to the promoter or more efficient elongation from the pausing sites. As we have seen the transcriptional upregulation after AGO2 knockdown, we used a ChIP-seq dataset from the ENCODE database and searched for AGO2 binding to the transcription start sites. Indeed, we found a lot of genes had TSS AGO2 binding peaks, and we could classify the genes into two classes. And you can see for genes with TSS AGO2 binding peaks, they show a significant trend of upregulation after AGO2 silencing. And by visualizing the binding pattern of AGO2 around TSS, we could see, very interestingly, AGO2 binding is not centered, but right downstream of the TSS. So we hypothesized, potentially AGO2 could interact with initiated transcription rather than helping initiate their transcription. So we further calculated the pausing index of each gene using another Grosik dataset, which could reflect the extent of transcriptional pausing of each gene. And we saw stronger AGO2 binding to the transcription start sites, stronger transcriptional pausing. So it's highly possible that AGO2 could serve as a transcriptional repressor by arresting the RNA polymerase from elongation. We further validated this mechanism using short-term for SU labeling and full coverage RNA-seq. You can see for highly paused genes that show upregulation after AGO2 knockdown, their nascent RNA coverage showed a 3N spike compared to control cells, which indicates more efficient transcription to the end of the genes. However, for highly paused genes that show no significant changes, their nascent RNA coverage just resembled between two groups. So we identified the non-canonical role of AGO2 in transcription using PerturbSci-Kinetics, and we validated this mechanism. Then we switched to the second question. Can we identify key factors of nuclear and mitochondrial mRNA processing? By using the same method, we checked the RNA processing status of single cells reflected by the nascent exonic release ratio and the mitochondrial transcriptome turnover reflected by the mitochondrial nascent reads ratio. We found knocking down OXPHOS genes and metabolism-related genes resulted in impaired RNA processing and mitochondrial transcriptome turnover. As mitochondrial transcription generates polycistronic transcripts, which is a very different mechanism from the nuclear RNA processing. So we can see knocking down spliceosome genes only has specific effects on the nuclear RNA processing, but has no effect on mitochondrial transcriptome. Here, we found knocking down LRPPRC has the strongest effect on mitochondrial transcriptome turnover, which is even stronger than direct perturbation on ETC complex genes. So we compared the steady-state expression, the synthesis, and degradation rates between the non-target cells, the ETC complex perturbations, and LRPPRC perturbation. We found cells can still compensate after ETC gene perturbations, as they showed relatively stable steady-state expression with slower down synthesis and degradation rate. However, knocking down LRPPRC disrupted the homeostasis. We could see a dramatic reduction of the steady-state expression level of all the mitochondrial genes, and their synthesis are highly impaired, and they have highly accelerated degradation. We also observed a similar result from another data set using LRPPRC knockout mice model. So our result is conserved cross-species. And by using another data set, we found the increase of secondary structure of mitochondrial mRNA after LRPPRC knockout positively correlated with their impaired RNA stability. So LRPPRC could open the secondary structure and stabilize the mitochondrial mRNA. As the functional consequence of this highly disturbed RNA dynamics, we also observed highest integrated stress response score in those LRPPRC knockdown cells, which is due to the highly functional defect of mitochondria. In our validation experiments, we also observed very well separation of two cell populations in the UMAP space, indicating the shift of cell states. So we identified LRPPRC as a unique mitochondrial mRNA dynamics regulator by using PerturbSci-Kinetics. Finally, we try to answer the third question. Can we distinguish the synthesis and degradation-driven gene regulations after the perturbation? We checked the RNA dynamics of differentially expressed genes between each perturbation non-target cell pair, as they are differentially expressed. So in theory, it could be driven by synthesis or by degradation or by both. Across perturbations, we found a median of 30% of differentially expressed genes were regulated by degradation. And by ordering these perturbations by using the number of synthesis and degradation regulated D genes, we found very strong enrichment of mRNA decay and mRNA processing genes in the plot. I'll show you some examples. So these genes are members of nonsense mediated mRNA decay pathway. These genes encoded RNA exosome. These genes are responsible for mRNA adenylation, and these genes are related to 5' mRNA cap. Besides, I would like to highlight two genes here, DROSHA and DICER1. These two genes are members of microRNA biogenesis pathway. After the transcription of microRNA gene, the prime microRNA is cleaved by DROSHA, and the cleaved pre-microRNA is exported to the cytoplasm by exporting 5, encoded by XPO5. Then these molecules are further cleaved by DICER1, and the stem loops are removed. Finally, only one strand of this double-stranded RNA is loaded onto the RISC complex for mRNA silencing. So DROSHA and DICER1 are two key RNases in microRNA biogenesis. By checking the RNA dynamics of their differentially expressed genes, we found more significant overlap between the degradation-regulated D genes in two groups. And it's clear that almost all the degradation-regulated D genes we found in dicer1 knockdown cells could be found in DROSA knockdown cells, which is very consistent with their positions in the pathway. As DROSA is upstream of dicer1, knocking down DROSA might have more fundamental effects. By further plotting the steady-state expression and synthesis and degradation rates of these overlap genes between different perturbations, we found, though all these genes showed steady-state upregulation, only this group of genes showed the activated synthesis. And for this group of genes, their upregulation is due to the impaired degradation. And to further validate their impaired degradation is related to microRNA ablation, we further checked the binding of RISC complex to their transcripts. So for degradation-regulated DE genes, you can see very strong binding pattern of RISC to 5' and 3' untranslated regions, which are exactly classic microRNA binding sites. And for synthesis-regulated DE genes, they showed almost no pattern. So we could confirm that this group of genes are regulated by microRNA-mediated mRNA degradation. For validation, we established single gene knockdown had inducible Cas9 cell lines, and we induced the knockdown, labeled the nascent transcriptomes individually. After profiling these cells with a larger cell number and with more sequencing depth, you can see well separation of these cell populations in the UMAP space, further proving the single cell resolution of our assay. Consistently, while knocking down XPO5 showed mild changes, we could still see well separation between the AGO2 knockdown cells and dicer1 and DROSA knockdown cells. By further calculating the rates of these candidate genes that I showed in the previous slides, we could see very conserved post-transcriptional regulations in dicer1 and knockdown cells. And for XPO5 perturbation, they showed milder but still similar responses. But for AGO2, almost no degradation changes were observed in this group of genes. So this matches our finding in previous. And to further validate the accuracy of our equation, we perform long-term for SU labeling and then wash off those labeled mRNA to directly track the degradation of mRNA in the cells. Again, we saw very strong consistency between different strategies. For example, for this gene, we almost saw no degradation changes after the perturbation. But for these two genes, you can see much higher RNA stability after DROSA knockdown. This results further validated our finding. So in summary, we developed a low-cost, ultra-high throughput combinatorial indexing single-cell CRISPR perturbation platform, enabling the co-capture of 3 layers of information. And we showcased the power of PerturbSci-Kinetics of identifying global RNA dynamics regulators and separating the transcriptional and post-transcriptional regulations upon the perturbation. Our manuscript is now on bioRxiv. We would really appreciate your attention. At last, I would like to say thanks to my PhD mentor, Jing, and every member of the collab. Thanks Flow Cytometry Center and the HPC Center at the Rockefeller University. And thanks CEGS for giving me the opportunity to be here. I will be very happy to take any suggestions or questions. Thank you so much.
Hi, thank you so much. That was very exciting. Let's move on to our Q&A. If you have a question, you can raise your hand or type it into the chat box. And we'll start with a question that is in here right now. So LRPPRC mutations in patients causes Lee syndrome and mitochondrial complex 4 deficiency in most tissue, as well as complex 1 assembly deficits in skeletal muscle. I was wondering if you were able to distinguish the different mitochondrial complex deficiencies in your results.
So I think from our result, we just observed a general compensation mechanism. It seems the RNA dynamics can still keep the stability after knocking down single EGC complex genes, but maybe the combined perturbation could cause more severe outcomes. Great,
thank you. Another question that I have here is, How much does the labeling of nascent transcripts improve estimates of cellular states over techniques like RNA velocity?
So I think they have different application actually, because for RNA velocity, it's more used to infer the cell state. And also we have different assumption. For example, for RNA velocity, it assumes all the RNA have mostly the same splicing rates. And for us, we did not really calculate the splicing of the RNA. So I think for us, we mostly focus on the synthesis and degradation rates calculation of the RNA. So we infer the kinetics of RNA rather than inferring the cell state transition. And I think we also tried using the RNA velocity method, and it was not as accurate as the metabolic labeling. So I think the metabolic labeling still outperform the RNA velocity at the current stage.
Okay, great. Thank you for that answer. If we have, here we have one more question here. Would you have expected more overlap between Drosha, Dicer, and AGO2 in their gene expression responses? Further, do you think you could infer which microRNAs were used in the AGO2 peaks derived from the eCLIP dataset?
So I think because AGO2 family has two proteins, AGO1 to 4. So I think the result that I show here is just because AGO2 is kind of redundant in the risk complex. So if we just knock down AGO2, maybe other AGO protein could still compensate the function of AGO2. So that's the reason why only more RNAi unique mechanism just stand out in our screen experiment. If we could knock out 4 proteins at the same time, I guess it will show us much stronger RNAi related results. And for AGO2 peaks in the ECLIP dataset, I think I tried to refer to some databases, but it's hard to tell because there are so many diverse microRNA binding sites prediction algorithms. So currently we did not really try to dig into that part, but maybe we could follow up later.
Well, great, thank you. Well, with that, I'd like to say thank you again for this talk. It's been very exciting. And to everyone, this seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. You can also follow us on Twitter at segs_ICA or check our website for the latest announcements regarding upcoming talks and events. With that, I'd like to thank everyone for joining, and we hope to see you at our next seminar. Have a lovely day.